Welcome to our newest episode of the Lebanese Physicians Podcast. And today we will be discussing the genetics and molecular mechanisms of autism spectrum disorder. And our guest is Dr. Maria Chahrour, who is a tenured associate professor at the Eugene McDermott Center for Human Growth and Development, the Department of Neuroscience, the Center for the Genetics of Host Defense, and Department of Psychiatry at the University of Texas Southwestern Medical Center. She's also an investigator at the Peter O'Donnell Jr. Brain Institute. She obtained her undergraduate degree from the American University of Beirut and subsequently did her master's at the University of North Texas in forensic genetics, and then at the Baylor College of Medicine for her PhD. She worked at Harvard University as a postdoctoral fellow for a number of years and then moved to UT Southwestern afterwards. Maria, thank you for being on the podcast.
My pleasure. Thank you for having me.
Yeah, Maria, I reached out to you. I was looking at LinkedIn and I found your profile to be interesting, especially with the research on autism spectrum disorder, which is big these days. So can you tell us first a couple of things? One is, how prevalent is autism spectrum disorder and how do you define it?
Autism is actually pretty prevalent nowadays. It affects about 2.8% of children in the United States. That's around 1 in 36, based on the latest statistics from the CDC. And there are more males affected than females, on average about 3 times more. The international prevalence, according to the WHO, at around 1%, but it varies substantially across countries and regions, depending on how many studies have been done and the rigor of these studies. So it's anywhere from less than 1% to over 3%. So autism is more of a spectrum of neurodevelopmental conditions. It affects how we interact with society and the environment. It's characterized by impaired communication skills, social behavior abnormalities, restricted interests, and repetitive behaviors. So these are the core symptoms, but in addition to these core symptoms, there's a host of comorbidities or conditions that also are frequently seen with autism. And they can be, anywhere from medical, psychiatric, behavioral, or developmental. Some common examples include intellectual disability, seizures, anxiety, and GI abnormalities. As I said, it's a very wide spectrum. And so every individual with autism is very unique. So if you've ever met one, you've only met that one person with autism.
Exactly. And that was my that's why it's called autism spectrum, because there's a spectrum of disease from what we call quote unquote mild, right, to more like severe impairment of social interactions.
Yeah, significant impact on the quality of life.
And given this, it's 1 to 3%, which is, I think, a high prevalence compared to some of the other, let's say, rare diseases that we talk about in different topics. But so what is its impact on society, the individuals themselves and their families?
It poses significant societal and economic burdens. It's actually more than the cost of cancer, heart disease, and stroke all combined. It's forecasted that the annual direct medical and non-medical costs on society are going to be around $460 billion in a couple of years. So as you mentioned, it's a spectrum with individuals who have mild symptoms and those with more severe symptoms. They may have, difficulties developing and maintaining friendships and relationships, communicating with their peers and adults, understanding what kind of behaviors are expected from them in a school setting or on the job. For families, addressing the needs of an individual with autism can be super stressful and puts them under a lot of stress, not just financially, but also emotionally, sometimes even physically.
Exactly. And I guess the cost that you were talking about is in the United States only, right? Not like internationally.
Yeah. It's in the United States only. Yeah.
And one question I have before we move on is the prevalence higher, let's say, in certain countries than others? And what would this be related to if the prevalence is different in different countries?
Yeah, so unfortunately, there are a lot of countries where, there's the entire continent of Africa, for example, prevalence studies have only been done in 4 countries. So, they kind of tend to aggregate in North America, some in South America, and in Europe, Australia, So I think the prevalence really varies dramatically. And there are some countries. So in the United States, the CDC study that I referred to is based on over 220,000 children, for example, And then there's other prevalence studies. For example, in Lebanon, the prevalence right now is around 1.5%, but the study is based on less than 1,000 children. So I think we need a lot more prevalence studies, but it's safe to say it's somewhere between 1% to 3%. There are some populations that potentially have higher prevalence because of specific genetic risk factors that predispose to this. And that's actually one of the active research areas in my group. There's some evidence to suggest that individuals of East African ancestry are at a higher risk, about 2 to 3 times higher risk than their non-East African peers.
Okay, we'll talk about that. Yes, this is a good segue into my next question to you. Is there a genetic component to autism spectrum disorder? If so, how much does it play a role? And is there an environmental component too?
Yeah, so autism has a very high heritability of up to 90%, and this is based on large twin studies that have been conducted over the past decade. What does that mean? What it means is that the proportion of the phenotypic variance that's due to genetic factors is about 90%. So in other words, changes in your genes account for whether you're going to have autism or not. There is a very small environmental contribution that's been particularly more challenging to pinpoint to prove actual causation versus correlation, because everything under the sun has been correlated one way or another with autism. You know, the mom was too stressed during pregnancy, she took ibuprofen, she took antidepressants, and so on. But actually, there is no causative role that's been demonstrated for any of these potential environmental factors. I can give you one example of an environmental factor that has actually been shown to contribute to autism risk, and that is the paternal age, so the age of the dad. The older the father, the higher the risk of these, what we call de novo or spontaneous mutations that arise in the gametes of an older individual to cause autism. So there's a higher likelihood the older the dad is. Of course, you know, increased risk doesn't necessarily mean that it will happen, but it does substantially increase risk. On the genetic side, as I mentioned, there's a very high heritability of up to 90%. And so far, hundreds of genes have been identified. So I would say around 300 or so with high confidence. About 3% of these genes are associated with very well-defined syndromes. For example, Fragile X, Rett syndrome, tuberous sclerosis, Angelman. And then there are about another 5% of cases are because of big changes in DNA. So we call these chromosomal abnormalities or copy number variants, CNVs. So these are either deletions or duplications of specific genomic loci. There are lots of hotspots where this tends to happen more frequently than other places, like the 16p11.2 locus or chromosome 15 Prader-Willi Angelman locus. So those are the two categories. And then the other really big category of genetic causes are these de novo or spontaneous point mutations. So these are just changes in a single nucleotide. It could be de novo or it could be inherited recessively or otherwise. And then finally, some cases, all of these that I mentioned happen in the germline, in the blood, right? So, but there are some cases that are due to what we call somatic mutations. And these are the ones that occur only in the brain, for example. So, you know, hundreds of genes have been identified, but for the majority of cases, so all of this that I just mentioned accounts for about 35% of cases. So for the majority of cases, the underlying genetic causes have not been identified. And because of this, you know, each gene that's been identified to date contributes to a tiny, tiny proportion of cases. So I'll just give an example of the Fragile X syndrome gene, FMR1, which is a top contributor. It accounts for only 1.94% of cases. And then there are genes that account for 0.08% of cases. So you can essentially start thinking about autism as this collection of individually rare genetic subtypes or individually rare disorders.
Exactly. And that could potentially be the reason why it's a spectrum and not like a disease that identifies all individuals in the same manner.
Exactly. It's so heterogeneous phenotypically as well as genetically.
Now, since we talked about the genetics, can you tell us a bit about the work you have done so far on the genetics and molecular mechanisms of autism?
Yeah, so my lab is interested in finding autism genes and identifying them, and then in studying their function in more depth in the brain. So we can find specific molecular mechanisms that they regulate, and then use this information back for diagnostics and the design of therapies. So there's right now a big burden in the diagnosis side of things, right? It's done clinically through these questionnaires. It involves intensive time from the family side and the clinician side. So a molecular genetic diagnosis is something that we're working towards. And then the design of targeted therapies in the future. We focus on the more severe genetic subtypes that have these debilitating comorbidities that I mentioned. And then I guess I'll talk about both the gene identification and then the more mechanistic side of things. We have two approaches that we use to identify genes. Our primary approach is by doing whole exome or whole genome sequencing. So this is essentially, the exome is the coding portion of the genome. And so we can sequence all of that or the entire genome. And we do that in familial cohorts. These are families who have one or more children with autism. And we focus on shared ancestry and ancestral diversity. So what shared ancestry means, basically, you can think of it as families where the mom and dad are related, either as first cousins, second cousins, or third cousins. And then there are entire populations that you can trace back to a common ancestor. So we leverage that in doing our gene mapping. And then the ancestral diversity refers to specifically our work on the African cohort that we have enrolled in our studies. And that's basically African populations have the highest proportion of genetic diversity in their genome, and they've been grossly underrepresented in these genomic studies. So that's something that's one of our main focus in the lab. So far, since we started in 2015, we've enrolled around over 3,200 individuals in these autism genetics research studies. And we have individuals along the spectrum of autism and from many ancestries: African American, Hispanic, East Asian, South Asian, and white. And it's an ongoing effort. We've identified several new genes so far that, we're focusing on a subset of those for our mechanistic studies. I guess I'll talk a little bit about our second approach, and please interrupt me. I know this is I wasn't sure how heavy I should go on the genetics or how light.
No, it's good so far, I think.
Okay, so the second approach that we use is something called forward genetics, and we do that in mice. So we mutagenize the entire genome of the mouse, and then we screen them for what we call autism-like behaviors. If we find a defect in these behaviors, we go back and we map the gene. And of course, the most important and final step in this is to validate the relevance of our findings to humans. And we do that by going back and screening for mutations in these candidate genes in clinical cohorts. So the idea here is that you can, the human genetics side of things is forward genetic screen in humans that was designed by nature. But the one that we have in the lab, the forward genetics in mice, is more efficient. We can do this in a high throughput manner. And at some point in time, we can hit every single base pair in the genome and what we call, saturation. So that was the idea. And we use these approaches together to support each other.
Yeah, so just to simplify it, in mice, what you're doing is you're causing the mutations, right? Yeah. And then seeing
randomly,
checking out if mice exhibits any autism spectrum behaviors, and then you go back and you find which mutation caused these behaviors.
Exactly. And then you also have to kind of validate to make sure, introduce this targeted mutation to make sure it was that particular mutation and so on. But the most important thing about our work is that once we identified these genes through these two concerted efforts, we're really interested in understanding their normal function in the brain and whether we can pinpoint specific mechanisms that they regulate, so we can, leverage that for targeted therapies. I can give two examples. We have, we're focusing on several genes, but one of them is a chromatin remodeler called KDM5A. And it works, its enzymatic function is to demethylate a specific chromatin mark. And when it does that, it affects the expression of the genes that it's targeting. So this is a gene that we identified as an autism gene. And then we found where it binds at these specific regions in the genome, and that it actually silences hundreds of other genes in the hippocampus and the cortex. So it's like a master regulator. And then, through other experiments where we can sequence individual cells in the brain, we found that actually when you lose KDM5A, there are very specific cells in the hippocampus that are super vulnerable to that loss. So now we're following up on that finding, okay, why is it required there? At what time in development it's required? And lots of other fun questions.
Basically, to summarize it, it's more of a gene regulator, I guess, right?
Yes, and exactly that. It's a switch.
And even a simple switch could affect a huge number of cells in the hippocampus, which could lead to autism spectrum behaviors.
Yeah, exactly. So we actually looked at around 100,000 cells in the hippocampus and found that very specific subtypes of excitatory neurons, not all excitatory neurons, but just some specific ones, and a specific subset of inhibitory neurons are affected. And there are other excitatory and inhibitory neurons and also glia and other non-neuronal cells that are just fine, they're doing fine in the absence of this chromatin regulator. So it was amazing that, when you lose it in just these specific subtypes of cells, you have such dramatic phenotypes on learning and memory. We looked at the hippocampus because of its connection to learning and memory. The other gene that we're working on, we're a little bit closer to translational studies. It's called UBE3B. It's mutated in a severe neurodevelopmental disorder with autism. So these patients have complete lack of speech, intellectual disability, and other features, muscle hypotonia and GI abnormalities. This gene encodes for an enzyme that regulates the levels of other proteins in the cell. So it's like a quality control system. So if it finds defective proteins, it tags them, and then the cell knows, okay, now I have to get rid of these proteins. So we identified this gene, and then we found a specific substrate that it's regulating, and that substrate is actually a master player in several metabolic pathways. I think the most famous one is branch chain amino acid metabolism. So you might have, know about it from, maple syrup urine disease and other neurodevelopmental disorders. So when we lose the gene, that metabolic substrate that we found accumulates in tissue, in the liver and in the brain. These are like and skeletal muscle. And so because it was being studied in metabolism, someone has actually developed an allosteric inhibitor for this. And so now we're injecting our mouse model with this inhibitor to see if, we bring down the levels of this metabolic regulator, can we rescue any of the phenotypes we see in the mouse model? So this is like more closer to translation, and it's an example of trying to repurpose a drug. So our hope is that if we study these specific mechanisms that are affected, we'll be able to pinpoint targetable pathways like this one that we can then harness for the development of personalized therapies.
Yeah, so I guess to make it simple, it's like similar to what we're doing right now, let's say because I'm a pulmonologist, basically what we're doing now for lung cancer, let's say, so identify certain pathways and you do targeted drugs for these different pathways, and probably a similar thing.
Yeah, actually, I love to compare autism to cancer. So, when cancer first started, I don't know, a long time ago, people thought it was one big monster, but then they realized there are different types of cancer. And then they started understanding the genetics of these different subtypes of cancers. And that's how, they got a handle on personalized targeted treatments, right? So I think we're just lagging maybe a decade behind, but we hope that, when we understand these mechanisms for these specific genetic subtypes, it'll help us usher in the era of treatments for autism.
Right. Because currently, just to summarize to people, currently, there's no specific drug therapies for autism. It's more of a clinical diagnosis, not a genetic diagnosis. And then you send...
it can be. So there are, depending on insurance, health coverage, and depending on the clinician, they might order in a clinical exome done. And in those cases, if it's something that's been already established, this gene, this particular variant or mutation in the gene is known to be pathogenic, then you can have a molecular genetic diagnosis. But because autism is so heterogeneous, and we don't think of it as this, right? We don't have the full catalog of genes or mutations yet, but we're getting there. Yes.
Yeah. So similar, so basically, for example, cystic fibrosis or primary ciliary dyskinesia, you can send like a genetic panel for that. And what you're trying to reach, the same thing for autism spectrum disorder, is we'll send a genetic panel to see if they're a carrier of some of the major or minor genes that are causing it.
Exactly. Yes. Exactly.
And that will hopefully have an impact on therapeutics. So currently for people like who have kids with autism spectrum disorder, what's your advice to them, I guess?
First, early diagnosis is very important because there are some behavioral interventions that can be done. And it's been shown that the earlier these interventions are started, the better the outcomes. Of course, the outcomes are very variable, unfortunately. So that's number one. Number two is finding out the genetic cause. Getting a genetic or molecular diagnosis will be important on many levels. So one is it answers a major question, right? Like why does my kid have autism? Two, it's important for genetic counseling and family planning. What are the chances that I'm going to have a second kid or a third kid with this condition? And then it connects them to other individuals who have mutations in this particular gene. So other families or a network of families that are affected by that particular genetic subtype. And that's important because there are lots of foundations now that follow these cohorts, like a natural history study. What's the prognosis later on in life and so on. And also to connect them to clinical trials, because right now we're at a very exciting time for translating our understanding of autism and neurodevelopmental disease into the clinic. So there are ongoing clinical trials, and even if not now, there will be because of several strategies that are being tested. So I think it's important to get connected to your specific cohort who are also suffering from the same genetic defect.
Clinical trials for therapeutics, how far away are we from those?
Yeah, so currently, like in addition to behavioral therapy, there are also treatments for comorbidities, like the seizures, for example. And so it's management of symptoms. But in terms of other treatments, there's a pipeline of gene therapies that's ongoing, and but there are also a lot of challenges associated with that. And the reason is too much of these genes can be as detrimental as having too little. And so in gene therapy, you have to make sure you're introducing the correct dose, correct copy number of the gene, and also to the right place, right? So the right neurons have to have this, or other cells in the brain, they need to have the right amount of the gene. So there are a lot of challenges associated there. But there are also studies looking at something called antisense oligos, or ASOs. And those allow you to kind of fine tune, like if you want to turn it more on, like the second copy of the gene, we have two copies of every gene, one of them is affected. Maybe you can tune the other one up a little bit to compensate. Maybe the mutation is a gain of function and you can turn that off. So it allows for very precise control of these gene levels. So that's a very hot area of research right now. And then ultimately, it's going to have to be through mechanistic understanding of these genetic subtypes. And not just, for personalized therapies, but also think of it like, you can start grouping them by these subtypes have a common defect in a specific circuit that needs to be addressed, or even brain region, and so on. There is another drug for a severe form of autism called Rett syndrome that's currently, just got approved by the FDA, and also manages the symptoms of Rett syndrome. So there is hope. I think, we're at an exciting time for these rare neurodevelopmental diseases.
And just to remind families, I guess I would just want to give examples. So because it's a spectrum, there's some mild to, let's say, severe behaviors or diseases that occur with it. But there's well-known individuals, I guess, who are on the spectrum who have succeeded significantly, right?
Yes. So I think, we on the research medical side of things, we are focusing on cohorts who have these more severe symptoms with really debilitating comorbidities, but there are individuals who are, their quality of life is better and they're doing well. Yeah. So absolutely.
Right. Exactly. Yeah. Anything else you want to add as a final words, Maria?
No, I think, we're hoping to with our research to generate enough momentum to start this self-propelling flywheel, going all the way from gene identification to translating it back to the clinic one day.
Yeah, so just to summarize, it's an exciting time, I guess, for autism research at this time, because it's turning into more of trying to find the genetic causes of the disease and molecular mechanisms, and then hopefully soon in the future be able to find therapies that target some of these genes and change the outcome for these patients.
Yeah.
Yeah, thank you, Maria, for all you do, and it was great talking to you today.
Thank you for having me.